These are the recorded lectures for Chapter 27, Part A, the reproductive system. And Part A focuses on just some general information about the reproductive system of humans, as well as how eggs and sperm are formed through the process of meiosis and spermatogenesis and oogenesis. And then it's going to focus on the male reproductive system specifically. Then when we get to slide set B, it will focus on the female reproductive system and then diseases like STDs, that kind of thing. So the reproductive system does not become active until puberty. The male and female reproductive organs are different, but they share the same four tasks. They form specialized cells called gametes, and the male gametes are sperm, and the female gametes are eggs or ova. So one of the tasks of the reproductive system is forming specialized cells called gametes. The second is bringing gametes from the male and female together through sexual intercourse or copulation. Oh, sorry. Okay, and then the third task is combining genetic information contained within gametes through fertilization. So fertilization is when the sperm fertilizes the egg and produces a zygote, which is the first cell of the new individual. And the fourth task is supporting the development of the fetus, which is called gestation, and the birth of the baby, which is called parturition. So gestation and parturition. All right, male and female reproductive structures are homologous structures. The male testes and the female ovaries, for example, the male penis and the female clitoris. So this just means that they share a common origin. The primary sex organs, which we call gonads, are the testes in the male and the ovaries in the female. And the gonads produce gametes and sex hormones. In the male, the gamete produced is called the sperm, and in the female, it's called the ova or the egg. And the process that forms these gametes is meiosis. And the sex hormones or steroid hormones are testosterone in males and progesterone and estrogens in females. And that will be one of the main functions of this chapter is learning about the different hormones and how they regulate the reproductive cycle in males and females. So the control of the hormones is the hypothalamic pituitary gonadal axis, the HPG axis. And it's the connection between the hypothalamus, the pituitary gland, and the gonads, whether testes or ovaries. So it's the connection between them. The hypothalamus will release hormones called releasing hormones or inhibiting hormones. and they will regulate hormones that are released from the pituitary gland, like FSH and LH. And those hormones will regulate the release of testosterone, inhibin, estrogen, progesterone from the gonads. Okay, so that is the HPG axis. And these are the main hormones, but what's not listed here is also the female hormones, estrogen and progesterone. And this is kind of how it works. You have the hypothalamus in the brain. Let's say it releases GnRH, which is gonadotropin releasing hormone. That will trigger the anterior pituitary to release LH and FSH. LH is luteinizing hormone. FSH is follicle stimulating hormone. And those will cause the gonads, testes or ovaries, to release sex hormones and inhibin, which will act on target cells. So at puberty, the hypothalamus begins releasing GnRH, gonadotropin releasing hormone, in a pulse-like manner. And that causes the anterior pituitary to release FSH and LH. And we will go into a lot more detail about what those hormones do. But just for now, I just want you to be aware of what the purpose is of the HPG axis. That's how the processes that affect reproduction are regulated. Okay. Now, meiosis is cell division of the sex cells to produce gametes. So meiosis is the process that produces gametes. And this is more of something that you would learn in biology, but it's important to the process of sperm formation and egg formation, and it's also information that you might find on the T's test if any of you are taking the T's test. So it's important to go over it. In meiosis, we'll have terminology like sister chromatids, which is when your chromosomes are replicated so that there are two copies of each chromosome that are identical. I'm looking for a picture. Okay, so if you look here at the cell that's going to divide and produce gametes, eggs or sperm, this is the original diploid parent cell that's going to either produce eggs or sperm. It's going to start out, in this example, it only has 2 chromosomes. Of course, humans have 46. Okay, so there are 2 chromosomes here. They're going to replicate, and they are going to have, each of these are going to have sister chromatids. They're going to have duplicated chromosomes, which consist of, they look like the letter X. They consist of 2 sister chromatids attached together. Those sister chromatids are identical. Okay, so then we have homologous pairs of chromosomes. So for every one of our 46 chromosomes, 23 came from our mother and 23 came from our father. So for each one from our mom that matches the one from our dad, they are homologous pairs. And by match, I mean that they look alike and they carry the same genes. Now, the chromosome that you inherited from your mom carries the same genes as the homologous chromosome you inherited from your dad, but they may not be the same versions of those genes. So they may both have the genes for height or eye color or skin color, but they may not be the same color. They may not be, one of your parents may be shorter and the other one taller, but the genes are the same. The genes, they're for the same traits. Okay, then we have non-sister chromatids. These are chromatids that don't belong, that don't are not found on the same chromosome. In other words, and I'll show you a picture that makes it a little more clear. During meiosis, when the homologous chromosomes pair up during prophase 1. This is going to happen during prophase 1. Okay, so let's say the green one came from your mom and the blue came from your dad. Okay, the two green strands are sister chromatids for the green chromosome. The two purple strands are sister chromatids for the purple chromosome. But the green and purple strand that are side by side, one coming from the mom and one coming from the dad, these are non-sister chromatids. And sometimes they actually will cross over and exchange pieces, which increases diversity. So, in fact, we can look down here at metaphase 1, see how there's a purple chromosome at the top paired up with a green chromosome. Well, the two purple strands are sister chromatids. The two green strands are sister chromatids. But the purple strand and the green strand that are side by side each other, they are called non-sister chromatids because they're not going to be 100% identical. They will have genes for the same traits, but they may have different versions of those genes. So, and diploid means that we have two of each chromosome, so that the diploid number in humans is 46. And the haploid number is 23. So in other words, we have 23 is haploid. Haploid is half of the chromosomes. So the haploid number in humans is 23. Sperm have 23, eggs have 23, and when they fuse and form a zygote, the zygote is diploid and has 46. The sequence of events in meiosis they're called prophase, metaphase, anaphase, and telophase, and each of these occur twice. As far as an overview or summary of what happens in meiosis, there's an interphase at the beginning, which is when your chromosomes are replicated. Your DNA is replicated, so you go from having one chromatid in each chromosome to having two sister chromatids in each chromosome. So during interphase, all chromosomes are replicated, and you have each chromosome has two sister chromatids. Then the first division starts. It's called meiosis 1. And during meiosis 1, you start out with 46 chromosomes, and this number is reduced to 23 chromosomes. It's the reduction division of meiosis. And then during meiosis 2, the haploid chromosomes, which consist of sister chromatids, those sister chromatids separate. So during meiosis 1, basically, the wording would be the homologous pairs separate, and during meiosis 2, the sister chromatids separate. During meiosis 1, you start out with a parent cell that's diploid or 2N, and the daughter cells, you end up with two daughter cells that are haploid or N. So they go from having 46 to 23 chromosomes. But those 23 chromosomes have two sister chromatids. So during meiosis 2, the sister chromatids separate, so you end up with two cells that have 23 chromosomes that are duplicated. Those two cells divide, and you end up with 4 cells that still have 23 chromosomes, but they are only consist of one chromatid rather than two. So you can look at the pictures. Here's your parent cell before interphase. This one has only 2 chromosomes. That's the diploid number. And it replicates, and you've got 2 chromosomes with 2 sister chromatids each. Still diploid number. After meiosis 1 occurs, you will have 1 chromosome that is still duplicated. After meiosis 2 occurs, you will have 1 chromosome that only has a single chromatid. So you start out with the parent chromosome is diploid, or the parent cell is diploid, and then the daughter cells that form are all haploid. There are 4 daughter cells that are produced, and they're all haploid. There's a process that occurs in meiosis 1 during prophase 1. It is called synapsis, and that is when the homologous pairs find each other and pair up. And when they do that, they form tetrads, because each of the chromosomes has 2 sister chromatids. That makes 4 total chromatids. Okay, and then the non-sister chromatids, one from mom and one from dad, go through a process called crossing over. They exchange pieces, and that increases the variety in the genetic traits that are passed on to the offspring. So after meiosis II occurs, you end up with 4 cells that each have the haploid number of chromosomes in them. So you would end up, for example, with 4 sperm that each had 23 chromosomes rather than 46. And you have animations that you can watch on meiosis. I encourage you to watch that. We will start with the male reproductive system in the next recording, and this will be slide number 37. So we'll start with slide 37 in the next recording.